Okay, I think by, by my watch, we have 2:30 Eastern Time. So respecting the need for people to be a part of this call and then move on to other business, we should probably get started. I'd like to welcome everyone again to the call and thank you for participating. I think we have a real interesting and valuable 90 minutes ahead of us. For those that don't know me, my name is Matt Kleinhens. I'm a vegetable extension specialist at Ohio State University. I work from my office at the Oso-Agricultural Research and Development Center, which is where I am currently. And my job over the next 90 minutes is to ensure that everyone on the call is repaid for their very valuable time, repaid for being here, and repaid in different ways. That repayment would come, hopefully will come in the form of information, but also the opportunity to share a question or a comment that's very, very important to you. So we want to make sure that we have time for everyone to say something that's important, but also to listen carefully to what others are sharing as well. So we're focusing on crop biostimulants that share three important characteristics, and I just want to set that stage. And for people who have joined us for the other two calls or listened to the other two calls, you might be recognizing that I'm going through a bit of the preamble that I do on each of the calls, which is important. And so for those that are familiar with it, I ask for your patience. But the characteristics of the products that we are focusing on include the following. One, they contain microbes as an active ingredient. Secondly, they are OMRI listed, and they are used by sustainable organic growers. And in addition to being OMRI listed, or instead of being OMRI listed, they may be listed by a certifying agency or organization. And finally, they are advertised to enhance growth and or crop resistance to stress, as opposed to, for example, what we discussed in the first call, biopesticides, which have a different role. Finally, I said there were three characteristics, but that was a bit of a fib. There's fourth. They're applied to vegetable crops, and that has been our focus. And for the purposes of this particular project, we are focusing on vegetable crops, although knowledge or information, insights, experiences with other crops can certainly inform our conversation. So do bring those up if you think they will be relevant. So before we get down to business, let's cover some background. Ground rules for the call, only being two. Please mute your phone if you are not speaking. You might recognize that from time to time there's background noise at your location, and please respect the call by muting your phone during that time if you're not speaking. And I'll just I'll mention that typically a person cannot hear the background noise at their own location, so they may be unaware that the background noise at their location is coming through to the rest on the call. Second ground rule would be to listen carefully, but please speak up when you are asked or when you feel the need to. We have a panel of resource people, and I'll introduce them here soon. But in truth, everyone on the call is a resource person. So please ask a good question, or share an experience to your liking or a point of view. Finally, if you would like, you can email us while we're live, or by us I mean me at this point in time, at vegbiostimsferts@gmail.com. I gave that email before. It's a rather long one, but it is vegbiostimsferts@gmail.com. That's a way to reach me directly about this topic, and when we proceed to the next stage of this project, that will be an email that you can use to reach all the project personnel. So unless there are any questions or concerns about the ground rules, we can proceed to the next stage. Okay. So a second bit of background. It's important to acknowledge the support that has made the call and the project possible. And that support is coming from the USDA Organic Transitions Program and the North Central SARE Research and Education Program. And it's also coming from the companies and organizations represented by all the people on the call. So I very much appreciate that support, and it's to everyone's benefit that it's available to us.
And would you repeat that first one? That
Sure.
First one again, sir?
In terms of the support for the call?
Yes.
Sure. USDA Organic Transitions Program, USDA NIFA Organic Transitions Program. That's the granting program for primarily transitional organic growers. So it's that Organic Transitions Program and the North Central SARE, Sustainable Ag Research and Education Program.
And do they carry online a list, I could check myself, but do they carry online a list of pubs and publications and studies and such and programs like this?
Yeah, so USDA Organic Transitions Program, what you would find there if you went to their, say their website, what you'll find most reliably is a list of the projects that they have supported through the years and links to those projects, rather than the resources that you name specifically, in abundance, okay?
Cool.
That's in contrast to SARE, where if you do find yourself at the North Central SARE or SARE overall, because there are region-specific versions, I guess. If you go to the North Central SARE website, you will find it to be more comprehensive than the USDA NIFA Organic Transitions Program, because they're entirely different. There at SARE, you will find links to resources, publications, webinars, field days, newsletters, and so forth. Okay, that's a good question.
Awesome.
Appreciate
Very
that.
Good.
Yep.
So this is Mark. I'd say that the
e-organic. Yes. Is there another question?
Well, you were talking about resources. The organic resource page of the extension website, developed by e-organic, is also a good place to find a lot of results, especially related to OREI and org.
Oh, absolutely. So, if it's okay, I would like for you to hold that thought and hold that message until we get into the substance of the conversation today, because we will be talking about tactics and steps that people can take as investigative teams and growers to get important information in the hands of people that can use it. Okay, and you've just named two, which is great. So let's table that for the moment and get some background taken care of, and then we will get into the real substance of the call, and I'm sure that will come out. Okay.
Sure.
Just for clarification, I know that a number of people introduced themselves as they came onto the call, but I'll just summarize quickly. As far as the registrants are concerned anyway, we had registrants for the call that represented 13 states, coast to coast and north to south, truly north to south and truly coast to coast. And they also represented different universities, different manufacturers, individual farms, and different consultancy businesses. So that's a pretty impressive group overall, and really grateful to see that. And almost everyone who registered expressed a specific example of how they are directly involved in sustainable organic vegetable production in some way. So if you were not able to introduce yourself to the group as you connected, and you would like the group to know who you are as you speak up, please do introduce yourself and tell us where you're from or who you're with, and help us to understand you that way. So unless there are questions, that would complete most of the background before I get to introducing our topic and our resource team today, the people who have committed anyway to sharing, and hopefully all of you will share as well. All right, so any questions so far, or shall I get right to the topic?
I would just introduce myself as Bob Rawson, but I'm the president of Bioremediation International, and I do quite a bit of work in South America in this area, which has fewer limitations. And my business partner, Nancy Vatikatakis, is listening in but not commenting.
Okay. Good to have you both. So our topic today is more or less a single topic, has multiple faces, but it's more or less a single topic. And the topic is prioritizing tactics for creating new and useful and research-based information, and then getting that information into the hands of people who need it, specifically to increase their success with products that I described earlier, microbial, biostimulants, biofertilizers. So we'd like to identify tactics for creating new, useful, and research-based information. We'd hopefully like to, if we can, establish a bit of a hierarchy or a priority list of tactics for accomplishing those two related, integrated goals of creating information, but also getting it into the hands of people who can actually use it. So that's going to be the focus of today's call. So all of you can add to that in different ways, but we do have so far, I believe, 3 people on the call who are committed to being resource people, and we'll hear from them at some point, I'm sure, during the call. The first is Dr. Bonnie Ownley. She's a plant pathologist with the University of Tennessee. We also have Dr.
Hi.
Carol Goland. Sorry, Bonnie. Sorry, thank you. We also have Dr. Carol Goland, the Executive Director of the Ohio Ecological Food and Farming Association, and she's on the call, I believe, from Columbus.
Yep. Hi, everybody.
We have Bruce Caldwell, the CEO of 3-Bar Biologics, and Bruce, I'll let you explain a little bit about what 3-Bar is before we move ahead.
Okay, hi, this is Bruce Caldwell. Glad to join you. So 3-Bar Biologics is commercializing beneficial microorganisms that were originally discovered out of the Department of Plant Pathology at Ohio State. These organisms tend to be gram-negative bacteria, which a lot of research has occurred on these types of organisms to demonstrate their benefits, but very few of them have been commercialized because when you take them out of their natural environment, they tend to die off rather quickly. And so we're working to address that problem. Right now, the product we've commercialized is in corn and soybeans, row crops. But we're quite interested in expanding to other higher value crops as well. So glad to be with you.
Great.
This is Subbu Kumarappan. I am listed as a resource person, but I'm here more to learn from everybody else. You all have more experience than I do on the biological products, so I want to listen and learn from you. And I do have some information that I would like to get some comments on.
Subbu, you're not going to get off that easy. Okay.
Don't know. We'll see.
Super glad you're on the call, Subbu. And that was Subbu Kumarappan. Subbu, if it's okay, I'll introduce you as an economist and educator. And he's with Ohio State University, and he's with the with the Agricultural Technical Institute, which is adjacent to OIRDC. 2-year programs there, and ATI does a phenomenal job at what it does, and we're very pleased to have Subbu not only on the call today, but also on this project to help people get at the financial aspects of the use of these products. Okay, so we would have had look forward to having Dr. Brian McSpadden Gardener, Sustane Natural Fertilizer. He was unfortunately called away to other business to another commitment, so we definitely will miss him here today, but look forward to hearing from him again. So that's why his name was listed at the website as resource person, but unfortunately he can't be with us. So again, we're gonna discuss tactics for creating info and getting it in the hands of people who can use it. That's going to be what we focus on this afternoon, or this morning, I guess, the midday for those on the west. And we'll hear from Subbu, Bonnie, and Carol, and Bruce soon. But I'd like to take a different approach to start out the conversation. I'd like to hear from other people, whether, Dave, I don't want to pick on you as a grower, or Bob as another manufacturer or somebody else, but I'd like to hear from somebody else besides the resource people, just at least initially, about this topic. This is certainly a first opportunity to comment. I think, in the spirit of what Subbu just said, to learn, I'd ask, how do you currently select products? What do you do when what's the best step most important step you take when using them? how do you evaluate their performance? What tactics or resources do you rely on? What seems to work, what doesn't seem to work so well? And I don't have anybody in particular in mind, so the microphone will be whoever shouts out first about how they have come to select, use, or evaluate products.
If I may speak briefly, Bob Ross.
Sure. Mm-hmm.
We have been involved in using bacteria, particular strains of bacteria that are similar to what many of the manufacturers use, the Bacillus subtilis, Licheniformis, and Pseudomonas fluorescens, for maybe 25, 30 years in wastewater and soil bioremediation. And so we developed our product around that, and then developed some patents related to growing those organisms in situ and in field applications for remediation. And it's sort of anecdotal things that we noted were like improved growth of vegetables and things like that in the treated soil. So in the course of one of the patents that we developed, growing bacteria to reduce the cost of application in the field, we included plant and animal applications, which set and sort of languished for several years before we actually got into the idea. Said, yeah, this is a better market and application than bioremediation, because people care more about food than they care about waste. But that's the brief of our entry into it, and we're trying to develop that market like everyone else, I think.
Okay. Bruce, you mentioned anybody else from that who is not a identified resource person. Anyone else would like to chime in on their selecting, using, and evaluating these products? That language, by the way, select, use, and evaluate. That language is very specific, because it is the language that I've heard growers and extension and consultants use with me often through the years. People don't know how to select a product or what product to select, and there are many. We established that earlier in this conversation way back in January. They're very confused at times about how to use them properly. The instructions on the label can be very can be inadequate. And then finally, as far as understanding or identifying what effect they may have had on the crop, it's not always clear on. They are not always clear on how to do that. So that language is taken directly from growers and other extension people. Those are three distinct processes. Do we want to tackle the selection first? We talked about that a bit in the first call. Are there any recommendations, Bruce or anyone else, that you would offer in the way of selection? You know, if you have somebody call you cold or email you cold, or you're just talking to say, hey, I grow vegetables. What kind of biostimulant, biofertilizer might outdoors? Is there a group of products that I would pay attention to most? What type of product would I pay attention to most? What would I be looking for?
Yeah, well, I think what Bob just mentioned was quite interesting, that the same organisms that might provide, say, some bioremediation to, say, clean up a waste material or decompose a waste material might also have plant growth promotion benefits as well. And I think that's not uncommon, that, there are many different services that these different organisms can provide. And frequently the chemistries that a certain organism produces can have different benefits, whether it's growth promotion or stress reduction or even a biocontrol, antifungal type of benefit, that kind of thing. So there's a wide variety of organisms that are being researched, both at universities and within companies. And I wouldn't necessarily prescribe one specific one, or I don't have a magic bullet for sure.
No. So here's the concern that I have, or a concern that I've heard expressed. There are currently roughly 173 products that fit the description I used up front. OMRI listed, biostimulant, biofertilizer, applied, potentially useful on vegetable crops, okay, and contain microbes. That number is larger than most crops that one finds in a very significant seed catalog, and certainly more than most varieties. What are we going to be in a position to do as a research extension and consultancy community and manufacturing community? What are we going to be in a position to do to help people make sense of those 173 and growing options? Is it going to be a completely wild west, or will there be guidelines? I mean, that's probably more. Yeah, I'll just put that out there. What's going to happen? What's going to be different in the coming years that will help people use these products more effectively? In earlier calls, select products more effectively. In earlier calls, we heard about how important it is for people to develop a relationship with their supplier. I don't think they have time to develop relationships with 85 suppliers. So what's going to change?
It might be good to have a matrix of things that are known that various products fall into, so that people could more rapidly narrow down to like 5 for what they're going after or what they're interested in, as opposed to having to go through 150 or so and growing. There'll probably be 1,000 before we're done.
Possibly. Other ideas or suggestions? Dave, I don't want to pick on you as a grower. I'm tempted to just ask, how do you select what you use? If you're willing to share.
I kind of fell into it by dumb luck, man. The same year I ordered a 3-bar product, I also had Jang and Julie come out and field test several other varieties.
Okay. So
several other strains of bacteria.
Yeah, right. But would you, again, Dave, I don't want to put you on the spot, and I know you'll you'll divert me whenever you need to. But is that a sustainable approach? I mean, could you do that like year after year?
No, I think moving toward where we're, the topic of the discussion, I would say that in the future, a matrix system that's geared towards specific vegetable crops would probably be the ideal approach.
Anything at all that can speed the selection process. Ones that are higher priority versus ones that are lesser priority. Is that right?
Yes, and like in my situation, growing roughly 40 different vegetable crops in a season, it would be helpful to know which products are specific to each, variety of crops.
Right. So as so looking to a manufacturer, supplier, or even somebody else for information, is that one aspect that you'd be looking for? Some sort of tool? I don't want to call it tool or resource that helps you separate the 10 that you should be most serious about from the 163 that you don't need to think about?
Yes. Yeah. It would save a lot of time, narrowing down.
Right on.
Specific species of bacteria. And also, soil types. You have to throw in, the matrix would have to be geared toward different soil types because, we've got, as you know, in Ohio, we have mostly clay soil, and other states have different soils. So there's no one-size-fits-all.
Right. And I think we covered part of that in I think it was the second call, the site specificity or the nature of the site influencing the possible outcome. Anything else?
There's some potential for looking at the how rapidly the bacteria might grow, and what bacteria it likes to grow with, and what environments and that sort of thing. Whether there's any pathogenicity with, like if you're doing multiple crops, some might be very good for one thing, but might actually stunt something else by affecting, say, the fungi in the soil in some way. While it's doing a benefit, it might be doing a detriment, for example.
True.
So there's those things, and whether they are known bioactive metabolites, things that are known about some of the species. They know more about like Bacillus subtilis than they do about almost anything else, or as far as the human body, they probably know more about the genome of some of these bacteria. So you can predict what they will do based upon the enzymes that they produce.
True. Dennis, if I could tap you a bit, you've had a lot of interesting points to make on in this general area about helping people select the right product for their application, and then we can talk about the use later. But would you like to add anything to this part of the conversation before we move on in terms of product selection?
Well, a little bit. We're talking about specific functions of the beneficials within that soil environment. And I think one of the things that we're missing that a lot of these beneficials have multiple functions within that soil environment based on need. So what we need to look at is not so much specific beneficial doing one job, but how does it function based on whether it's stress, based on whether it's drought resistant, nitrogen fixing, what benefit does that specific strain, which a lot of times when you look across the board, they have multiple benefits depending on need. And I think that's some of the information that needs to get out there too. I mean, so often we look at a specific strain of like Bacillus subtilis, and we don't think of all of its different functions that it might be doing within that soil environment. Are these mycorrhizal helper bacteria? Are they drought resistant? Are they phosphorus solubilizers? What all are they trying to do? And I think sometimes with growers, they need to, and it comes down to what we're doing right here and now, getting more information out there so when they go through and have an understanding of what these different strains can function or what their functions can be within that soil environment, and then go after exactly what they're trying to achieve. But so often we find that, diversity is the key in a lot of cases based on need of the plant of what it may need in a given period of time.
True. So Joseph, again, I'll tap you if it's okay. Specific to mycorrhizae.
Sure.
I was told very recently, and again, this is just me parroting what somebody shared with me, is that mycorrhizae may be less useful in an annual cropping situation, like I'll mention Dave's annual vegetable cropping, unless they're applied each year. The soil levels are kept at a functional level, and in addition to that, they may be even more helpful or beneficial if paired with bacteria. Is that your perspective as well?
Well, I would say everything depends on certainly the growing conditions, because, it's well known that, fumigation reduces the population of the natural soil microorganisms, including mycorrhizae, And so if you have an annual crop where you use fumigation, or if you apply high levels of fertilizers, particularly nitrogen or phosphorus, you certainly reduce the natural level of mycorrhizae. So it is recommended to reapply every year. And certainly, if you have an annual crop where you remove the crop from the soil, that will certainly remove part of the mycorrhizae as well. So every crop needs to be inoculated, I would say. As for the combination of mycorrhizae with bacteria, I don't have any direct experience with that. I know that we have products that contain bacteria package and mycorrhizae together, and growers seem to be very satisfied, although I don't have a direct comparison of pure mycorrhizae or mycorrhizae with bacteria. I think it depends on the crop and the needs, or what the goal, what the purpose of using not just the mycorrhizae but the soil inoculant. What is the condition that you try to improve? So there's not a general kind of recipe.
Can't make a general recommendation at this point, at least your perspective. But what I heard,
yes.
Part of what I heard in that last section, I apologize. Part of what I heard in that last section is speaks to the single microbe inoculant versus the multiple. I call it microbe. I mean single species. Obviously, that's not the right word all the time, but it's bacteria only, fungi only, or bacteria and fungi together, because that is a way that growers and others can use to differentiate the large number of products that are available. Bacteria only, fungi only, or bacteria and fungi. And getting back to Dennis's point about the diversity factor, if you will, and the many roles that an individual microbe can have. I mean, are we in a position where we are becoming more serious about mixed inoculants, or not?
I think that's a good approach, and there's some studies. This is Bob Rosson. In Florida on the citrus greening, which is not a vegetable crop, as you know, but that they found that when the number of not just the population, but the diversity of the population of some of the important soil microorganisms falls below 3, that the crop exhibits the signs of the citrus greening, even though it can be asymptomatic and still have the disease and last a long time with that disease, maybe 5 years. So the diversity, there's good evidence that these guys work together and help each other and protect each other, particularly like the bacillus protecting the Pseudomonas in drying conditions. Those kinds of things are important.
Okay, so we may not be in a position here as this group to necessarily offer a recommendation. So that's not what I'm going to do here next. But I would like to consider encouraging everyone on the call plus others to obviously keep up the good work that they're doing in that way and drive towards something that might resemble a statement like, when in doubt, choose a mixed inoculant. When in doubt. There are steps that a person could take to have obviously be more effective than that. But if the science is leading us to say when in doubt use a mixed inoculant, okay, that's a recommendation that somebody could use to kind of cut through some of the chaos, if you will.
There's one thing.
Go ahead.
There's one thing that I've found that's true, and you could probably this as well. Is time of year of application. The various plants have have needs that change over their growing period. Phosphorus at one point, nitrogen at another, those kinds of things. Plus the soil moisture in California gets dry in the summer. So if you don't get the bacteria down through the Grand Canyon of pore spaces to the root, you're wasting your time. You can spray it on the surface of the ground, it penetrates an inch, and you don't have anything. You need to have it get down to the actual root, and that usually implies getting it down there when the soil is wet enough to act as a carrier to the root.
Good point. I'll just make a quick reminder. When you're not talking, consider muting the phone. That way it'll eliminate the background noise at your place. Okay, anything else that we would like to cover at this time regarding the selection component? I know that we have not exhausted it in any way, shape, or form, but we are here to talk about...
Hey, Matt, this is Bruce.
Yes,
just one quick question on the idea of multi-species consortia. Because I agree with the logic that a broader diversity of beneficial organisms is better and could perform over a wider range of conditions. One of the problems, though, as we're kind of working on consortia relative to single strain delivery, it can be very difficult to provide good quality control and to maintain viable populations of, say, more than one strain as you go multi-species. That's one reason there's not a single biopesticide registered with EPA that's more than a single strain, because of that difficulty in quality control. So I wouldn't necessarily default to say that more is better, because that's not necessarily the case.
Fair point. And I'm glad you spoke up. This is the kind of calibration that is essential, I think. You can't let whoever it might be on the call let them go free, if you will. So that's I hope everyone heard that, and of course people who listen to the recording can hear it again twice. So we're not at the point where we can say mixed is best. I think we might be at the point where saying, as somebody did when they wrote in, we need more third-party validation of efficacy, or even first-party, manufacturing demonstrations of efficacy, as we talked about in a previous call. But many appear to be clamoring for third-party validation. And part of this project is designed to help with that we are not out here at all, and I'll explain a little bit later. This project is not designed to identify the best performing product from among those 173. That is absolutely not the objective. But the objective is to provide, one objective is to provide quality research-based information on a subset of products, and simultaneously help people develop a process whereby they do become more comfortable in selecting, using, and evaluating, most importantly, evaluating products on their own, because we do assume that farmers and extension people and consultants and, of course, manufacturers will need to be evaluating products on an ongoing basis. And so a process to do that effectively seems to be necessary. We do not have an IR4 program, for example, for biostimulants and the like. So anything to Bonnie, Carol, Subbu, anything to add on the selection side before we get to the tactics for use side?
This is Mark Shelbeck. A couple of thoughts, or maybe this is actually getting into tactics, but one is I was at a presentation on root health by Joel Groover. He's with the University of Michigan. But anyway, he talked about using products like these not as a bulk soil application, but directly applying to roots. I mean, the most obvious is when you're transplanting starts to, dip the root in your desired mycorrhizal or other inoculant. And the second thought I had about selection and the difficulty of maintaining the efficacy when you mix two different products, it's easier to make at the manufacturing level a single species product. You might then apply, as a farmer, you might choose to apply two products, one being a mycorrhiza and one being a bacterial, for instance. You get that diversity, and you get the presumably, if you have good products, you get this quality control as well.
Fair point. We might be in a position where people are buying and using multiple products. If the quality control for creating a mixed is difficult, and a mixed and a diversity is important, which I think at some level the jury may still be out on. But that's a good point.
Hey, Matt, this is Bonnie.
Yes.
I wanted to throw in a few thoughts. I think this has been a good conversation, and some important issues have been raised. I'm a big fan of root dips and seed treatments rather than application to bulk soil. I think that root dips and seed treatments are much more effective. So, and you and I can talk about that as far as some of the on experiment station tests we're going to do, because it gets the organism right to the root where it needs to be, so it can have a food supply from root nutrients, rather than laying out in bulk soil and maybe not surviving at all before a root grows towards it, because they're not going to move very much in soil. The interesting thing about multiple products, and the research is certainly out there, that there's value in multiple products, particularly some of the things that have been mentioned. The plant needs different things at different times, and these organisms have different functions. But there's also work that shows the combinations of these organisms, even the proportions of the organisms can be critical. So it gets a little difficult to just recommend multiple products. And I see when a grower, if they buy these products and then they're basically experimenting themselves with trying to mix and match and see what works together. But that information is something that's sorely needed.
Well said. I will just jump in real fast to say that, Bonnie, you referenced the upcoming experiments at both the University of Tennessee and at Ohio State. The plan, as folks, for other folks on the call, the plan as we now have it set is to use root drenches, root dips, or seed treatments essentially exclusively. At least we here at Ohio State have no plan to apply materials in any other way. And we will be applying materials to butternut squash, tomato, carrot, lettuce, and I know I'm forgetting something else. But yeah, so to Bonnie's point, we'll use root drench, root dip, and seed treatment, or seed treatment.
There is some good experimental data on that out there, on the number of colonies necessary to prevent a mildew on a seed.
Right.
You've probably read up on that already.
Right. So moving on to use, and I know we had a very active conversation about use over the previous two calls in some way. So we won't necessarily replicate that here, but within the context of developing information that we eventually want to get to people who can use it, are there specific recommendations or thoughts that people have about how to use the products in an evaluation? And I don't want to poison the well with ideas that or thoughts that I have had about this. I'd just like to get everyone else's thoughts first on using the products in an evaluation. And all of you on the call can take that whatever direction you want to take it.
Well, this is Philippe Douillet with Ecomicrobials, and I'm sorry, I would like to say a couple of things. The same way that with chemicals, we don't know sometimes how they interact together when we put them in an agricultural field. and the chemistry that is altered, and the manufacturers cannot tell you what happens when they interact with different molecules. In a very similar way, when we mix biologicals, we can get interactions that are not beneficial. I mean, some products that will be affecting the other products. So one recommendation is to do some in vitro bioassays, in which you can look at if some organisms will be antagonistic to others. And I think that that's a little work to do. It can be done by a lab, a microbiology lab, or a person that has a little bit of experience in the field. And that will give you already an idea that you are not killing one of your beneficials by adding the other.
Philippe, it's great to have you on the call. I'm so glad that you mentioned that. Yeah, Bonnie, were you going to say something?
Yeah, I was going to say something. If you're looking at these endomycorrhizal organisms, we're not able to grow them in culture.
Yeah, well, mycorrhiza are a little bit different from the bacterial products. I agree on some fungus. What we have seen with mycorrhiza, especially bacillus type of microbes, they enhance the mycorrhiza. They make a more favorable environment. Then I'm not so much afraid about the interaction between mycorrhiza and bacteria. I'm more concerned about different types, group of bacteria, because they are quite, and some fungi, free-living fungi that are producing some antibiotics, metabolites that can be antagonistic to each other. That's why my point goes. Okay.
Okay.
I'll put this out to the microbial ecologists among us. Okay, Philippe, Dennis, Bonnie, others. What you just said requires me to ask: if you look at the components in the 173 products on the market, is it possible that applying more than one product could actually be counterproductive? And do we have anything to offer growers toward saying these are bad combinations that don't work or don't live and play well together?
There's already research out there on bad combinations.
Okay.
So yeah, it's certainly possible that some of the combinations will not work.
Okay, so we would need to go back to the ingredients, if you will, of the products that we know about and scan for those bad combinations and possibly say, think twice before using product A and product D together. Yeah. Is that reasonable?
Well, one of the tricky things...
That would be useful information.
Yeah, okay. I agree, but there is a tricky part is the taxonomy. Taxonomy of microbes, it's in the infancy, and depending on the method that has been used, you get a completely different result. If you use lipid analysis, you get some classification. If you use a molecular, if you use a biochemical, then and some tests of these products were analyzed and identified the microbes many years ago. Doesn't mean that the taxonomy will be current with what it is now. On the other hand, for example, Bacillus subtilis, you will have like 50 in the American type culture collection. There are over 50 different Bacillus subtilis, and many of them are completely different one from the other. And when people say, "Oh, my formulation has a Bacillus subtilis," doesn't mean that it's the same that Joe Schmo that has also Bacillus subtilis. And that's what it makes it a little tricky just to go by the information on a name to say, "Oh, this should be working very well with the other." That's why I point out the need sometimes to or stick to a series of products that are known to be compatible to each other because there is some previous experience, or getting some evaluation of that if you're going to use, massive amounts. Or if you do one single application once in a while, probably it's not such a big deal. But if you have a recurrent program of rotation of products, that's what I'm more concerned.
That's a great point. A lot of these organisms, I think, were named prior to the big push to use molecular techniques to really pin them down as far as their taxonomy goes. And you're right, some of them could be, maybe they're not what they're were originally identified as. So.
Okay, Bonnie, help, and others again, help lead me through this if you don't mind. I'm still compelled to ask, would that confusion within the taxonomy more or less nullify the potential value of a table or chart or something that says these may be incompatible with each other, or would that just be a time not well spent?
If I might chime in briefly, it might depend upon, or it will depend upon if you're applying to soil in a dormant state, like on a plant, preparation of a soil or whatever, as opposed to foliar application, because in the one aspect, that robust community of organisms in the soil is probably helpful, but in the other, its function to keep other organisms from colonizing is important, and so dominance of one or more species on the surface might be more efficacious.
Yeah, and also there's a point that has to be made when you apply. Also depend on the dosage rate that you apply to a field. It goes, for example, when you put a product in the soil. Yeah, it's a fraction of the microbes that are there, and I agree that would be an increasing in diversity, and probably the antagonism effects are not going to be so violent because there will be kind of a spreading effect of the microbes that you're applying to the system. When you're treating seeds or you're doing something like that, or in a nursery, you're going in a nursery where you apply high doses, which is relatively high doses of a particular biocompound, and then you apply another one, especially in effects of antagonism for control of diseases. That's what I have seen in the field, some the nullification, of the beneficial effects of one biological because the other one is completely antagonistic. Some trichoderma and some bacteria, for example, might be very antagonistic because of the type of antibiotics they will produce. Then if you come along with we have a disease and then you come along with two biologicals that are antagonistic to each other, you might get the disease completely, wiping out your field, and you're using two biocontrol agents that are very effective by themselves.
Carol, I'd like to take a different tack on this stage of the conversation. OFA was very kind to allow Julie Laudick and Brian McSpadden Gardener and I to get a better understanding of the use of microbials within the OFA grower community. We have reported that within that vegetable grower community, more than 40% have used a microbial inoculant each year for at least the last 5. And other market data indicate that the microbials industry is growing very consistently. There are many people making lots of money on these products. A question that remains is, are growers making money on these products as a function of their use? So Carol, if I could, you're listening to the conversation and you have your perspective. What do you think?
I was hoping Subbu would be able to tell us something about that. What I take away from listening for the conversation is that the evaluation of necessity has to be incredibly complex. The evaluation of the efficacy before we even get into any kind of return on investment. And so I'm not sure that our growers have the tools they need to make that determination about whether or not they are benefiting economically from the use of these products. Since we've heard consistently there's not a single magic bullet. We have to understand data that we generate about these products are going to be valid under a particular set of conditions and not others. This using these products really necessitates a commitment to a program, not just to a product. So I think we are at the various earliest stages of growers being able to make that evaluation. And maybe Dave will correct me on that. But there also, I think what we see in the organic system plans, you can kind of see waves of product du jour. Something comes into fashion and then it goes out of fashion and is replaced by something else. That very anecdotally suggests that as a collective, an evaluation has been made and people have decided to move on to something else.
Matt, I was going to add something to that. Carol just hit a great point that it's a program, and I think one of the things that is key to get information out to growers is the fact of the understanding that we are working with a living system here, and a living system needs to have food, it needs to have shelter, it needs to have water, it needs to be applied. I mean, there's so many beneficial products that can be used in the organic industry, but they are not going to function as designed if they do not understand the environment that they are putting them in and making sure that they can grow those within that soil environment and keep them alive. And I think some of the, I guess I would say, failures are when they don't see the expected result that they anticipated to see based on the use of that product. Generally falls down to either misapplication or the wrong environment that we have placed those into, whether we're talking mycorrhizae, whether we're talking biology, looking at phosphorus levels when applying it, or applying mycorrhizae with phosphorus in furrow application. All that, it's correct. It's a program that needs to be designed in order for this to be functioned. And from my experience, I think that's the most important information to get to the growers also is how do we keep this stuff alive?
So,
amen to that. And multi-year in that program, rather than looking at just a few applications and hoping that everything's going to be hunky-dory.
Correct. I mean, biology does not, one application of biology is not going to create these unbelievable results that we hear out there in yields and building soil health and all these things. It goes back. It's a program, and it takes time. We didn't destroy this environment in one day. We're not going to fix it. It took years of agricultural practices to get ourselves into the position that we are in, and it's going to take a few years to build that soil back up. And having an understanding of how we need to do that, I think, is critical.
Could it be that matrix has limiting factors built into it that would guide a person who wasn't as knowledgeable in that area? Like limiting factors like how much carbon or how much phosphorus or whatever is available?
Correct. So your carbon ratio is going to be critical. Your organic matter is going to be critical. Someone talked earlier about time of year, application, temperatures, moisture availability. All of these things are going to determine how effective the application is going to be. And we talked earlier about root dip or seed treat. That is the key to this, is getting the inoculum in the environment that it needs to be. In furrow application. All of these are critical, is critical information for the grower to understand. And so often I see, and we've talked about this earlier, but I see labels out there that, people say you can tank mix it with other things. And so often we have to look at the concentration rate, both in organic and conventional agriculture, of what environment are we putting this living system in a tank, in a solution, and we expect it to survive. That to me is one of the things that needs to get out there. of the information. I always tell guys when they're doing biology, do biology. If you want to put a food source with it, great. Fish, kelp, humates, enzymes, they're compatible together, but don't try and tank mix things that are not compatible.
So the last 10 minutes or so have been particularly informative to me in this way, in the following way. What's been described is a situation in which growers have access, and their consultant, the people that they rely on, like Extension and others. It assumes that there's a lot of information available, and it also seems to be a good example of dilemma and opportunity being two sides of the same coin. In my opinion, all of the things that have been said here recently are entirely reasonable. The problem is, and they point to there being, you need to know this, 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 you need to know this and this and this and this, and take this and this and this into account. But as I look around, there is no research-based information in grower-friendly or extension consultancy-friendly form that, one, consolidates and integrates and synthesizes that information, and two, makes it widely available. So it seems like if I'm listening to this as a grower or an extension person, I would have to have a personal, professional, a relationship of some significance with a product manufacturer, which seems to be the primary reservoir of this information. I'd need to know Philippe really well, and he would be able to. I'd have to download his brain and to help understand and use his program, or Joseph's program, or Brian's program, whoever it is. That seems to be the situation that I see. Do others see it similarly?
I actually do a little bit. And this is Dennis again. One of the things that I look at is for someone to buy a product off a shelf or online and not have an understanding of how it functions and doesn't have the information themselves on how to use it is not going to perform as they intend it to perform. So often it takes a lot of time for quality companies that are trying to get these products out there to have an education with the grower that is using them to get that information. And I think part of that is because this is still relatively new to most growers. And as time goes on and that information and more, standards come out based on using these inoculum within the soil environment or as a foliar, it will be easier for the grower to understand. And that's one of the things that I see this conversation that we're having right now is to get that information out in a way that growers can, some kind of website or something that is easy for them to do to maybe get some of this information, which is just a light understanding of how to use these products and what is important when when using them in a given application. I mean, that's some of the things we're talking about right here.
Yes. Yeah.
Yeah.
And this is Mark. I just want to briefly note that I think we've heard some really pretty strong trends here that there already be a few general points. The whole idea about getting it right into the root zone or right on the seed, being careful what you mix it with. Obviously, don't let it dry out, freeze, or overheat. So you want to go on an evening of a mild, damp day and put it on, if it's going to be a field application. And there seem to be a few themes here.
Mark, I would agree. To that, I would add, seek out a company that can really counsel you well with, comprehensive information on how to use the product and what not to use it with.
Mm-hmm.
That'd be a fourth one, I guess, that I'd chime in.
Yeah. From economic perspective, this is Subbu, by the way. I have a couple of questions for the group here. From economic perspective, when I was searching for information on microbes, there is very limited information available there. There are two things. When we do this crop production budgets, we do not purposefully, we do not separately identify the microbial products or anything like that. And microbial inputs are rather really small compared to all other inputs. So my question for those who are looking at the yields and other things, can we specifically say that this much amount of yield increase or a quality increase happens because of microbes? That is my major question, because, from my perspective, when I am trying to quantify, when I am trying to calculate the economic value, that becomes important or huge for me, I guess. So any comments from anybody in the group would be useful for me.
I have one or two. First off, the land is your equity, and if you are building the soil, that's what a farmer should be doing long term. And so it's pretty good data on the fact that these microbes help the soil. So from a capital standpoint, you're improving your capital resource. That's a economy. But as far as on the shorter term, somebody else might have another comment on that. But it appears that this is more of a, when you're talking about the soil, it's long term. When you're talking about individual survival, then there is some data on that for seeds and such. And then there's, anybody else want to chime in? Go ahead.
Well, I use the example all the time that for too long we have been going to the bank and making withdrawal. It's time for us to make a deposit back into that soil environment. And the things that we're talking about here, and it's just not one specific product. It's, it's beneficial bacteria, it's mycorrhizae, it's trace minerals. It's a combination of things to go back into those deposits to that soil environment, which is going to give us that building of soil health. And we need to get those things all working together in order to get the end result, which I think the question is does it increase yield? And from my experience on a variety of different crops, I can say a good biological program, and when I say biological, I'm including a lot of things, it has shown better yields, better crop resistant, better soil building health, better organic matter. All of these things will come with the right program. And it goes back to what we were talking about earlier, is being able to get the right education to the grower. It's kind of like when we go to the doctor and he prescribes us some medicine. If we went there and just grabbed what we wanted and took it, we could really mess ourselves up. And that's what we're coming out here too, is we need to get the right prescription within the soil environment in order to get it to function as we intend it to.
Sure, that's a good
I know the other one thing I wanted to mention, I agree with you, is that there's how much you're spending on alternatives such as pesticides, herbicides, and fungicides, which comes on the cost side of the economics for a grower.
Well, and I look at that as not only the cost associated with a fungicide, an insecticide, or a pesticide, but each time we apply those, we get a one-time short-term benefit. When we're doing biology, we are looking at long-term benefits. So I've always looked at a insecticide or a fungicide as a rescue for a plant that is already stressed or not doing well based on usually a nutritional deficiency. When we look at adding a biological program, not only are we trying to build soil health, but we're trying to reduce our susceptibility to having to use these things. So I see it as a long-term investment rather than a rescue.
And it's also a mining operation to use those other alternatives, like for example, a fungicide that kills the mycorrhizae or the hyphae in the soil, and leaves it carbon deficient and changes its structure.
So Subbu, if I'm following the conversation correctly,
Sure.
it leads me to conclude that some unconventional, if you will, approaches may be needed to quantify the potential benefits, including like we've heard about. I call them offsets. So we use microbials and we don't need something else. Okay. And we can talk more in depth about each one of these topics later, and I know we will. But overall, it would seem like and I know you are familiar with this, so it's not something that you're learning for the first time, but a straightforward, more or less standard or conventional approach to the economic evaluation may not capture a lot of the reasons for which they are. Many would say they're used, or how they function.
Sure, that's true. By the way, just to, I found this one recently. These microbial products in agriculture, they are projected to grow at a rate of, say, 7 to, say, 12 or 15% even, depending upon the product. So, farmers are going to be adopting this in a larger manner, but then to understand whether it is economically meaningful, useful, and what is the application rate, and what should be the right product. These are the type of questions. And if this research can address that, I think it will serve a good purpose.
And that could be a whole other set of forums in itself, based on not only looking at the yield potential that you get from these, but all the other things that we have talked about here, and also even the longevity. I'm looking at we were talking the citrus industry or the orchard industry, of what is the longevity or the genetic potential of that plant to continue to give high yields, based on what we do based on overall soil health. So I think there's a lot of different things. We can't just look at yield. We have to look at the whole combination of a package of what these benefits are based on using them.
Thank you.
So therein lies one of the. Go ahead. Go ahead, please.
Oh, this is Steve at Tiny Biological. Just wanted to pipe in a bit, and we talked before, and a lot of times the trouble is we want to have a reductionist view. We want to find an individual organism. And a lot of times you look at the EPA biopesticide list, it is a specific species because we're looking at an individual antibiotic that's produced that has specific function against a fusarium or Phytophthora. But in a biological system, a reductive approach ignores a lot of our capacity. So if we're looking at bacteria with, say, an ACC deaminase pathway that's going to reduce ethylene during an abiotic stress environment, we're going to be ignoring a lot of times, this organism, like an azospirillum, is also producing nitrogen fixation, free-living nitrogen fixation, but on the other side of that, it's also a phytohormone producer. So a matrix approach and a pluralistic approach to looking at the benefits is going to be much more beneficial. And before a topic was brought up, the idea of plant specificity, I view this typically more as an enzyme pathway specificity. Do I need more nitrogen fixation? Most of these plants are going to be using these pathways in similar ways. So if I have a calcium triphosphatase that's produced to break apart phosphorus from calcium, that's not necessarily plant specific. That's more environmental specific. So a lot of these pathways, which was brought up before, and having the biochemistry and the bioassays available, that seems to be, from my perspective, a more beneficial approach than specific plant, specific environment. So just wanted to pipe in a little bit there.
And it's measurable. It's, whether or not an enzyme exists in a bacteria is a measurable thing. You could say it is or isn't in a particular strain.
Absolutely, and easy to do in a lab, much more difficult to do in a living field scenario where it's much more difficult to extract specific enzymes from a soil colloid, from a soil structure, soil medium, versus a soil-less medium Like a lot of times you'll see with the biocontrol, you can physically see a barrier and a suppressive ring around those organisms. That is difficult to do in a soil environment around a root in the rhizosphere. But we know these organisms, based on lab research, they have the capacity. The other side of this is we haven't really talked about root exudates and specificity of those root exudates. Jill Clapperton and other researchers are starting to look into this, but we don't fully know. They found a few molecules, malic acid, for example, that's spit out by the roots that helps encourage the growth of Bacillus subtilis when it is presented with a stress, typically with like a fusarium stress. The plant exudates will change based on the environment and environmental need. So the idea of diversity that Dennis has mentioned and some others have mentioned seems to be the most critical. I view these organisms as a Swiss Army knife. You can only use that tool if it's available on that Swiss Army knife. So multiple approach, fairly continual, consistent approach makes the long-term viability of these options more practical.
With regard to, like, for example, Bacillus, not Bacillus, but Pseudomonas fluorescens and some of the other pseudomonids that aren't as nice, but in fluorescens anyway, it is able to pick up plasmids that are already present in the soil or from cohorts, other bacteria that might be being grown with it. And I've used that for many, many years in bioremediation to try to specifically culture on site a better blend for that particular pollutant. So I think that might be applicable also to agricultural applications.
True. So in the Bonnie, are you speaking up?
Yeah, I disagree. I agree with him. That's a good point.
So noted. So I wanted to just briefly recap and then maybe transition. I'm noticing the time, and I've also made some notes about notes here specific to some of the comments. We've been covering selection, use, and evaluation, and understandably, we've covered the surface on each of those. It became clear to me, and perhaps to you too, that there's so much more to be done in each of those three areas in the way of research and outreach, positioning people to be better at all three of them. So, again, I truly appreciate what people have shared in terms of, guidelines, if you will, or ideas for selection, use, and evaluation. In the last few minutes that we have scheduled on this call, I'd like to share with you a more descriptively, I guess, more comprehensively, the project that binds Bonnie and me and Subbu and Carol and growers like Dave, but not only Dave, and manufacturers like Philippe and Joseph. I'd like to describe that project for you and also get people's perspective on the way forward. Okay, so before I get to the project specifically, Dennis, it might have been you who mentioned what we're doing in these forums, in these calls, and what possibly could be done going forward. I'd like to just put a question out to the group here. And I know that it's unique to us right now. I'd like to just put a question out to you and ask, and you can email, call, or say right now. Should we continue these forums in any way? Should these continue? Is there value in them? How would we maintain that value? That's something that I think the group would welcome some input on.
One of the things I keep hearing this conversation today about how we can help the grower understand how to identify and use these products. And I think for me, so often it would be great to have a group of growers on the same type of call-in that just ask questions based on how to help them identify the finding products. So get a group of growers in here and let everybody else just sit back and listen, and have those growers give us information that we could put out there to help them understand how to use biological products.
I couldn't agree more. Just as a matter of record, these conference calls were advertised nationally and very heavily within the north central region of SARE, but all nationally, with lots of different partners and through the extension networks and so on. And it was clear that, and directly to some growers, I mean large groups of growers. We had on the call who we had on each call. They're voting in some way with being on the call or not. We have had a great cross-section of the so-called, industry, if you will, but we have not had a very strong presence of growers, despite the fact that they have no reservation about sharing questions or concerns, in other formats. So I don't know if it's a timing, I don't know if it's an education like, hey, what really happens on these calls, or what, but I totally agree. I would really like to have more growers on the call.
This is Mark. Would it make sense? Oh, no, go ahead.
Go ahead, Mark.
I was wondering if it's worth maybe having sessions at conferences in different parts of the country. I mean, like when you have a conference, you get all these growers together, and as spring comes on, it's going to be harder and harder to get growers in the call because they'll be out there tilling the fields and keeping up with their livestock. And so I'm wondering maybe that's something to consider next winter. I mean, to have sessions where one or more people on the core, one or more core members of the team would be at a conference. I mean, like the Moses conference is one that's not too far from you, or the OFA conference.
Right.
I'd
that's a great idea. Yeah, that's part of the plan. That's baked. That's, if you will, that's baked in the cake, and we do plan that. But of course, the organizers have to agree. I did submit it as a possible idea for both Moses and OFA conferences. But that's part of the plan. But we can't just wedge our way under the program. We'd have to be invited.
We might throw Acres USA out there also.
Right. We will be authoring publications in various, like in Acres, like in TILF, like in association newsletters, like the OFA newsletter. We will be authoring publications, so we'll get the word out that way as well. So, but the calls specifically, the January to March time frame, or the December to March time frame. Do you see value in them? Overall,
this is fantastic.
Okay. So again, if you'll allow me, I'd like to summarize quickly some of the specifics of the project that have brought this group of people together, and thankfully have brought others to the table as well, who we hope to maintain very, very productive relationships with going forward. So this is not this project is open in the sense that we are aware that a lot of people can contribute to it, but the people on the project obviously have roles to play and things they must do, in other words, to satisfy the contract, if you will. So, as I mentioned up front, the overall effort is supported by both the USDA and SARE through the Organic Transitions and the Research Grant Program. We will conduct on-farm and on-station evaluations of somewhere between 4 and 6 products each year on vegetable crops. Those products will be selected in cooperation with our grower cooperators and the manufacturers, okay? We will, of course, conduct these independent evaluations here at Ohio State and University of Tennessee, and then, of course, with our grower cooperators throughout in various states, including Ohio and Tennessee, but not limited to those. We overall will follow guidelines for on-farm research that most would agree are, very, very reliable. But again, I'll say we'd like your input. Some of those guidelines will include SARE, Organic Farming Research Foundation, and other very well-known and respected guidelines for on-farm research. I don't have time to go into the details of right here, but I'd just like to mention those specifically. We will be very mindful of what people have said on these calls and what people have shared with us in other ways in terms of what are the components of a very successful evaluation and educational program. The team considers that we are working on behalf of our peers, research extension, consultancy, manufacturing, and growers. So we'll be very mindful of all of that when we are completing the evaluations. And this is my opinion, okay? I'm not speaking for anyone but myself when I say this, but I believe it has some general support throughout the team and throughout the group of people that I have corresponded with about it. We will evaluate products, and we will assist growers in evaluating products on their farm. Much like Dave mentioned earlier when he said he kind of fell into a project with Jung, who is sitting here across the table from me, Jung Wong, and Julie Laudick, who's now with OFA, a former grad student of mine. So we will assist growers in those evaluations. We very much want quality data because I think others want quality data as well. That being said, and we will aggregate those data, as the circumstances allow, to identify trends, like, hey, single inoculants versus, mixed and the like. But we are also, I think, very aware that what everyone gains in the process of evaluation might be more valuable than the outcome of any single evaluation. So the dialogue that we will have, the questions that we will address, open-ended, like we don't have the answer, so that's the next stage of work, or the information that we're able to transfer, whatever direction, I think is arguably more important, or at least as important as the outcome of any one evaluation that provides data on one or more products. So that's my thumbnail of the project, and we have just a couple of minutes left for those on the project or not officially. I certainly welcome your follow-up.
This is very good. I love this program. Any ideas about lobbying USDA to look at their regulatory methodologies for some of these ubiquitous bacteria and fungi that are non-pathogenic, and stop looking at them like they were chlorophos or something like that?
Yeah, I guess I would encourage you to, Carol, I'm going to tab you. I would encourage you, to follow up with your local grower association, follow up with the manufacturers. There is something called the Biostimulant Coalition. I can help with that, but I would encourage you to take the first step to explore who else is thinking along those lines, and then take a trip to DC if you can.
Done it before.
Sure. Yeah. It can work actually. So any other quick comments before I kind of wrap up with this set?
I'd just like to add that I have really enjoyed the conversation and am very much looking forward to this project.
We're getting
whatever
started,
we can contribute.
And that will be a lot. I'm certain of that.
Yeah. One last question from here.
Please.
If you want to share afterthoughts, which I probably will, the veggiebiostimferts@gmail.com, is that the right email address to use?
You can. Yes, that is the right email address.
Will that be shared with everyone?
I'm sorry?
Will those comments be shared with everyone on the list or available?
Yes, they will.
Great.
My intent is to follow up with everyone who has registered for the calls, whether or not they participated or not, and extend them. Of course, we have a listserv as well. You may or may not know that, but that's at the same Bugs in a Jug webpage. So a listserv, as I'm sure, some of you are members, but you can put a question to the listserv, and then people will respond in kind. That's one way to do it in kind of an open forum. The Veg Biostim's Fert's Gmail address, I can share the login information with whomever, would be appropriate, and that way everyone would see the email traffic. But to me, that would be repeating the listserv at some level. The Veg Biostims First, the Veg Biostims First Gmail address is meant to go to Bonnie and me and Subbu and Chris Taylor, whom we haven't mentioned, but is also a very important member of the team here at Ohio State. But that would go to all the quote-unquote collaborators on the project, and they would be able to respond to you rather than having you send an email to 5 different people. Okay? So I would use the listserv for a wide open
I joined the listserv a while back, but I'm not sure if I've gotten any emails on it yet.
Yeah, it's been kind of quiet, I think Jung has put a question out there or two, but, maybe we will ping the listserv with a message, and that may stimulate this. But that'd be one way of keeping the conversation active by email anyway. I'll just mention that we have recorded this call, and we intend to post it at the Bugs in a Jug main webpage. So if you'd like to listen again, or you think it would be useful to for other people to hear, please direct them to that website, and they can listen to the recording. And there is a tab underneath the recording to click if you find the recording to be useful. We're not asking you to, love us or like us. We're just wanting to know if the recording is useful. Look for us to follow up with emails, with other calls, with webinars, like I was mentioning before. And please do reach out to me, Bonnie, Subbu, Carol, anybody on the team if you have a question or comment. And unless there are other comments for the good of the order, we can wrap up.